In this last video, in chapter 25b, we look at digestion in the small and large intestine. So we're beginning here with digestive processes carried out by the small intestine. As you recall, this was a site where you had numerous organs actually contributing to the function. So you have the liver, the gallbladder, and the pancreas all depositing secretions here through this hepatopancreatic sphincter. And so the idea here is that first off, recall that chyme entering the duodenum is going to stretch the wall of the duodenum, and it's going to change the pH of the duodenum. And as we saw affecting the stomach, that was what caused the enteroendocrine cells, or the duodenal cells, to actually release secretin and cholecystokinin. Now in the previous video, we looked at the fact that both secretin and cholecystokinin slow the production of secretions from the stomach. And the goal of that was to temporarily inhibit gastric activity so that the duodenum had time to focus on this new chyme that had just entered. Well, your secretin and cholecystokinin also play a role here with the liver, the gallbladder, and the pancreas. Beginning here with secretin, we find that secretin can stimulate bicarbonate production from the pancreas. Now this is important because as bicarbonate ions in the form of sodium bicarbonate are produced by the pancreas, they can travel down here into the duodenum. Now bicarbonate ion, as we continue to review, is an excellent buffer. So it can buffer the acidic chyme that's now in the duodenum. Okay. Second thing secretin can do is stimulate bile production by the liver. So the bile here being made by hepatocytes is going to travel down through these right and left hepatic ducts and then here up into the cystic duct where it's stored in the gallbladder. And that's where cholecystokinin comes in. And so cholecystokinin finishes the job here by stimulating enzymatic secretion from the pancreas. So all these enzymes that are going to help you break down nutrients in the food, they follow that sodium bicarbonate through the duct and enter the duodenum. And secondly, you now see cholecystokinin causing bile secretion from the gallbladder. So the gallbladder begins to contract and the bile flows out through the cystic duct, down through the common bile duct, and now out through this relaxed hepatopancreatic sphincter. And so now all this chyme from the stomach is mixing with bile, sodium bicarbonate, and lots of enzymes from the pancreas. So let's look at what's happening with that pancreatic juice, as we will call it. So again, the sodium bicarbonate coming from that pancreas itself is going to buffer acidic chyme in the duodenum. Now you will recall that we had an alkaline tide of bicarbonate ions coming from the stomach after hydrochloric acid was made. Well, here now that you're making sodium bicarbonate, you actually have hydrogen ions that are produced in the blood as a byproduct of this, and it actually buffers the alkaline tide. And so the pH of the blood flows back in a more or less neutral sense. The pancreatic acini then are the ones that are making the rest of pancreatic juice. And this is going to be a combination of pancreatic enzymes as well as zymogens. You'll also find here that while this can be stimulated by the cholecystokinin from the duodenum as we saw, the vagus nerve, the same one that stimulated secretion of the stomach, can also stimulate the formation of your enzymatic pancreatic juice. Keep in mind, as we look at pancreatic juice, some of these zymogens are what we would see formed in an inactive state that then have to be activated. The other zymogen that we've looked at so far in these notes was pepsinogen being activated to pepsin. And so this table right here gives you a list of the different enzymes and zymogens from your pancreatic juice. So it starts here with your sodium bicarbonate to neutralize hydrochloric acid, and I left it in there even though it's not an enzyme or a zymogen. But here now you see your three zymogens: trypsinogen, chymotrypsinogen, and procarboxypeptidase. And so you can see each of those three being produced by that pancreatic duct: trypsinogen, chymotrypsinogen, and procarboxypeptidase. Now, since they're all inactive, they have to be transformed. And so right here, trypsinogen is going to be formed into trypsin. So trypsinogen cleaved into trypsin. Chymotrypsinogen becomes chymotrypsin, right over here. And procarboxypeptidase becomes carboxypeptidase. There we go. Even though there's more details that are given here, what's really important is that I think that you understand that it digests protein. right? So it's hydrolyzing polypeptides, it's digesting protein. This is very similar to what we saw pepsin doing earlier on. Now, in addition to that, you have enzymes that are being produced through this pancreatic duct, and these are actually secreted in an active form. And so the first is pancreatic amylase, digesting starch into oligosaccharides. So this is very similar to what we saw salivary amylase doing in the mouth before it was inactivated at the stomach. You also see pancreatic lipase, so digesting fats or triglycerides into monoglycerides and fatty acids. And then finally, you have your nucleic acid digestion taking place with ribonuclease and deoxyribonuclease, breaking those down into pentose sugars, nitrogenous bases, and phosphate ions. Now, this is really good. This is some really nice digestion that's taking place here, but it's not complete. So even though the pancreatic juice has begun to digest proteins, carbohydrates, lipids, and nucleic acids, these are not all small enough to be absorbed. And so you have to see one more level of digestion take place before absorption is capable. And that's where the brush border enzymes come into play. Now, these brush border enzymes are named for the microvilli or the brush border here on your enterocytes of the duodenum and the jejunum. And you can see here they are named maltase, sucrase, and lactase for those responsible for carbohydrate digestion. Maltase digesting maltose, which is a disaccharide, into glucose, which is a monosaccharide. Sucrase hydrolyzing the disaccharide sucrose into monosaccharide glucose. And lactase hydrolyzing your disaccharide lactose into glucose. The idea here is, once again, you are breaking things down into glucose itself because that is what's most readily usable in the body. You continue to see protein digestion take place as aminopeptidases and dipeptidases begin to break down those dipeptides and tripeptides into individual amino acids. And then finally, nucleosidases and phosphatases completing the digestion of nucleic acids. And so at the end of the activity of the brush border enzymes, the goal is that these individual monomers are ready to be absorbed. And so here, as review, which of the following secretions neutralizes the acidic pH of chyme entering the duodenum? Is it A, hydrochloric acid from the stomach, B, trypsinogen from the pancreas, C, bile from the gallbladder, D, sodium bicarbonate from the pancreas, or E, cholecystokinin from the small intestine? The next question, which of the following secretions chemically digest triglycerides into monoglycerides and fatty acids? Is it A, chymotrypsin, B, pepsin, C, pancreatic lipase, D, pancreatic amylase, or E, bile? The next question, which of the following brush border enzymes converts maltose sugar into glucose? Is it A, lactase, B, amylase, C, sucrase, D, aminopeptidase, E, maltase? And so, assuming that digestion is completed and all of these molecules are small enough to be absorbed, it's a new word, absorbed, you're going to see majority of that absorption take place here in the small intestine. So it's not just the nutrients that are being reabsorbed, though. You want to see here that the small intestine is actually the site of the greatest water reabsorption in the GI tract. So you haven't really noticed it, but nearly 9 liters of water are deposited into the digestive tract daily. So this is going to be water that you're drinking and eating, but also all those enzymes that you're secreting, those are mostly water-based. And so that's water that you're potentially losing and wasting from the body. Well, 8 liters of all of this is absorbed by the small intestine. We'll find that almost a liter, almost the remainder, is absorbed by the large intestine later. You can see 2 pathologic activities take place here when your water absorption is not appropriate. The first is diarrhea. So diarrhea occurs when the large intestine and small intestine absorb too little water. So typically this is associated with infection, and the body is trying to clear itself of the infectious organism. And so instead of slowing all these absorptive processes down so that water is absorbed enough, it rushes through propulsion and does not reclaim enough water. Constipation is the other extreme, and this is when too much water is reabsorbed. And what can happen in the large intestine here is that the feces or the fecal material becomes too hardened and is not very mobile. And this happens quite frequently with individuals who are dehydrated frequently. And so that's water reabsorption. Now, the small intestine is the greatest site of nutrient reabsorption, and the idea here is we want to remind ourselves that the macromolecules had to be digested into their smallest components first before this could take place. Second thing, and this is important for bile, is that only hydrophilic molecules can be absorbed directly into the blood. Hydrophilic mixes well with water, but if you start to think about lipids and lipid-soluble vitamins, those are hydrophobic. So you got to do something about that. And so that's where we talk about bile. So we said this at the very beginning of the chapter 25b notes, and we said that bile is essential for emulsification of fatty acids in the small intestine. So mixing with watery secretions. Bile is produced, then stored and concentrated and released by the gallbladder. And this is going to be stimulated in terms of secretin, stimulating the hepatocytes to produce it, and cholecystokinin, stimulating the contraction of the gallbladder to release it. Now, as you look at the function of bile, that's carried out by some of its individual derivatives. Bile salts are what allow the large fat globules to be emulsified. So if we take a look at this image here, you see the big fat globule from what you just ate, right? Think of butter, something delicious like ice cream and milk. All the bile salts right here that make up bile begin to essentially separate that fat globule into smaller parts. So it makes it a lot easier to work with something small now that all these regions are exposed to your watery digestive enzymes. The next thing here is that now those pancreatic lipases can begin to hydrolyze the triglycerides in these mini globules. And so you start with something slightly bigger here, and as those lipases begin to hydrolyze the triglycerides, they're broken down into smaller components, your monoglycerides and fatty acids. Now again, those components still can't just float through all this watery environment. So they have to be surrounded in little pockets known as micelles. Now, micelles actually are formed by what's known as lecithin. So lecithin most people have heard of. It's basically a group of phospholipids in the bile. And that group of micelles transfer all the fatty acids and the monoglycerides through the lumen all the way here to the intestinal wall. And this is where they need to make it in order to be absorbed. Now, at that point in time, they make it across the wall and are picked up here by chylomicrons. And these little chylomicrons right over here are combinations of these tiny little proteins plus the fatty acids and the monoglycerides. And those chylomicrons are how all these components are transported through the lacteals of your villi and all the way to the liver. And so it's important to know that bile is not just essential for emulsification, but know here how each component of bile plays a role. Okay? We keep going then and we take a look at what's happening with carbohydrates. So once again, larger carbohydrates need to be broken down into monosaccharides, the three of which are glucose, fructose, and galactose. Keep in mind that most of these monosaccharides are going to be absorbed across the small intestine and into the blood, but the large carbohydrate cellulose cannot be chemically digested and remains as is in the large intestine. And this is what we think of in terms of non-soluble fiber, right, or bulk in the large intestine. Now, 80% of that sugar that's absorbed is glucose. And this is going to be important because it has to be carried across these cells in a specific way. And so here you can see the lumen of the small intestine where all those individual monosaccharides are floating around. And here's the microvilli of the enterocytes. Now, fructose can be transmitted right through here through what's known as facilitated diffusion, going right through a protein channel, and then finishing up its transport right here into the core of the villus where it's picked up by the blood. However, your glucose right over there, as well as galactose, need to be transported through a symport system. Now, these are these little purple proteins. That's known as the sodium glucose transporter, and it's very reliant on a sodium-potassium pump. So the sodium-potassium pump is going to establish a gradient for sodium ions, and as those sodium ions follow their gradient into the cell, glucose follows them or galactose follows them. And so as long as your antiport sodium-potassium pump continues to run, you can continue to transport glucose into the cell. Right? And so that's the idea behind glucose transport. Now, the movement of these monosaccharides here into the core of the villus creates an osmotic gradient here. So this osmotic gradient that you see actually increases the water absorption from the small intestine. And so water here tends to be absorbed right there through what we call solvent drag, through leakage channels in tight junctions. And in some cases, you can have enough solvent drag that glucose follows the water. So glucose leads the way, water follows it, and some of the glucose dissolved in the water follows that. So it's a really nice positive feedback cycle. Absorption of proteins is really similar to this. Again, keep in mind proteins that have been hydrolyzed will eventually break down into amino acids. So here you see those tiny little amino acids right there hovering in the lumen of the small intestine. They are going to cross through the apical surface of this enterocyte using a sodium-dependent amino acid cotransporter. Really, this is just like the sodium glucose transporter. Here you see the sodium ion entering the cell, and the amino acid just hitches a ride and goes in with it. Right here, as you reach the basal membrane here of the cell, then this amino acid carrier for facilitated transport transfers the amino acid across and into the blood. When you look at nucleic acids, they tend to be pumped across the epithelium into the blood through membrane carriers that are similar to the sodium glucose transport. Vitamins themselves tend to be absorbed unchanged. They don't actually go through digestion the same way these other molecules did. In order for fat-soluble vitamins like A, D, E, and K to be absorbed, there has to be absorption alongside other lipids. And so there are individuals that if they don't see enough lipid in their diet, or if they have gallbladder malfunction and they're not absorbing lipids the right way, sometimes they end up with a deficiency in these fat-soluble vitamins. It's a little bit easier if you're a water-soluble vitamin, though, and these guys can be absorbed just through simple diffusion following solvent drag, essentially. B12 itself remains bound to intrinsic factor because it is so large and has to be brought across that way. Finally, of course, there's the absorption of minerals. So minerals themselves can be absorbed any place that water is. And so they can be absorbed along the entire length of the small and the large intestine. Some use cotransporters, like a sodium cotransport, and some use pumps where they go against their gradient. Iron and calcium are the only ones absorbed proportional to the body's needs. The rest are just absorbed freely. And so to end everything, quite literally, we reach the large intestine, the rectum, and the anus. So even though this is not the site of the greatest water reabsorption is still one of the most important functions of the large intestine. It's important because it allows for the compaction of feces, the absorption of water-soluble vitamins, and of course other things, some small nutrients that sometimes don't make it through the small intestine. It does take about 36 to 48 hours to convert all the remaining elements in the large intestine into feces, and so these do tend to take a little bit slower to pass through. Now, in addition to this, you do have a bunch of living creatures in the large intestine, and that's totally normal. And these are going to be known as bacterial flora. And so you might freak you out a little bit to think about the fact that bacteria are there. But if you've ever taken a probiotic, then basically what you're doing is you're trying to restore the bacterial culture of the large intestine. These are important for a number of reasons. They synthesize B and K vitamins. They can help metabolize a lot of these products like mucin, heparin, hyaluronic acid, and to some extent cellulose that your body has not been able to facilitate. They also can help to ferment indigestible carbohydrates. Now, of course, the downside of this is what produces gases. And of course, you do make sure that you have the right level of each one of these, and you do that by having housed dendritic cells within the wall of the large intestine to constantly sample these bacterial flora. And they regulate them through an IgA-mediated response. And as long as everything stays balanced, then this is a nice symbiotic environment. The problem comes when it's no longer balanced. IBD can begin to occur whenever the immune system flares up in response to bacteria that are present. And so even though something may not be harmful, these IgA-mediated responses can cause inflammation and pain and discomfort. Likewise, if you take an antibiotic that's powerful enough, you can actually wipe out the floral culture here, and you can see side effects of fecal production here in the large intestine. So we've talked a lot about the chemical regulation of digestion and absorption, but we haven't really looked at the timing of motility. And so this is where we finish up looking at regulation of the large and the small intestine, is with motility. So small intestine uses segmentation to mix food and peristalsis to actually move the food along and into the large intestine. The pacemaker cells in the small intestine are very similar to those in the stomach. They trigger contractions 12 to 14 times a minute in the duodenum, but it's a little slower in the ileum, about 8 to 9 times per minute. And so that creates a little bit of a backup in the ileum, essentially, until you open up the ileocecal valve to let chyme through. Peristalsis itself waits. It's initiated every 90 minutes to 2 hours and basically has a nice, what we call migrating motor complex that sweeps all the chyme from the duodenum all the way down to the ileum, pushing it towards the ileocecal valve. As you'll recall, gastrin in the stomach had the ability to mediate the gastroileal and the gastrocolic reflex. And this peristalsis is going to be initiated in part by that. And so it's a long reflex where gastrin creates this gastroileal reflex, increasing motility of the ileum and relaxing the ileocecal sphincter. As this occurs, your chyme can pass through the ileocecal sphincter and the ileocecal valve and enter the cecum of the large intestine. Now, anything in the large intestine still goes through segmentation through haustral contractions, but then it also experiences mass movements where you have a gastrocolic reflex stimulated by gastrin in the stomach once again. And so if you've ever experienced a situation in which you start to eat and then you realize it is time to defecate, that is because of gastrin in the stomach being stimulated by the stretch of the stomach, causing the gastroileal and the gastrocolic reflexes to begin emptying the small intestine and the large intestine. The defecation reflex itself is triggered by distension of the rectal walls. And so here you can see a couple of things. This is one example of a rectal valve meant to trap gas. And then here, of course, you can start to see the narrowing of the rectum and the anus itself. I want you to also see here 2 bands of muscle. So here you would see the internal anal sphincter on either side and the external anal sphincter there. Now, reflexively, what happens is as soon as there's fecal content here in the rectum and it distends the rectum, that causes the internal anal sphincter, again made of smooth muscle, to relax. Now, as that relaxes, it means that the rectum can actually dilate, allowing defecation to occur. But it may not be the time or place for that to work. And so that's why this outer band with the external anal sphincter is made of skeletal muscle over which we have conscious control. And so that's where you can actually regulate the timing of defecation to an extent, even though the reflex associated with defecation may have already started. At that same time, you can use skeletal muscles like the levator ani to lift the pelvic floor and empty contents of the large intestine. Likewise, Valsalva's maneuver, where you begin to increase intra-abdominal pressure, can actually help to expel contents through defecation. Last thing you can see right over here are the hemorrhoidal veins in this position, and those are actually clustered in this area but can begin to swell whenever you have too much strain or too much pressure here on the more distal region of the rectum. And that's what can cause hemorrhoids, which can be painful and which can actually rupture and bleed. And so I do want you to make sure you're familiar with the defecation reflex to an extent. Once again, you have voluntary fibers here going to your external anal sphincter with skeletal muscle. And here you have parasympathetic motor fibers running to the internal anal sphincter. Anytime there is stretch receptors right here due to feces here in the rectum, that is going to stimulate only the parasympathetic fibers to cause relaxation, but you have voluntary control here over the external anal sphincters to prevent that until the time is right. So last few review questions. Which of the following types of transport enables the absorption of monosaccharides, glucose and galactose? Is it A, solvent drag, B, transport through tight junctions, C, antiport through the sodium-potassium pump, and D, symport through the sodium-glucose transporter? Please choose the best answer. The next, which of the following types of transport enables the absorption of the amino acids across apical membrane surfaces of the intestinal wall? Is it A, solvent drag, B, transport through tight junctions, C, antiport by the sodium-potassium pump, D, symport through the sodium-dependent amino acid cotransporters, or E, facilitated diffusion? Question 13, which component of bile transports digested monoglycerides and fatty acids to the intestinal wall? Is it bile salts, phospholipid micelles, or chylomicrons? And that is it. You made it through the digestive system. I'll see you guys in lecture.